Welcome to another deep dive. You sent us a fascinating research article about, the role of a specific enzyme in breaking down DNA in our blood, and let me tell you, it gets really wild. Get ready to explore the world of plasma DNA fragmentomics.
It's definitely a cutting-edge area of research with some really interesting implications.
So before we get into the nitty-gritty, let's quickly define what plasma DNA actually is. We're talking about those little fragments of DNA that are constantly floating around in our bloodstream, right? They come from cells all over our body that are dying and breaking apart.
Exactly. And what's so cool about these fragments is that they're not just random debris. They actually carry a lot of information about what's going on inside our bodies. It's like a molecular fingerprint that can tell us about our health.
Okay, that makes sense. And the article you shared focuses on one specific player in this plasma DNA story, an enzyme called DNASE1L3. That's a mouthful. What makes this particular enzyme so important?
Well, DNASE1L3 is essentially a molecular sculptor. Its job is to carefully shape the size and sequence patterns of those DNA fragments in our blood. But the article goes deeper, looking at what happens when this enzyme isn't working properly.
And they found a connection to lupus, right? That's an autoimmune disease where the body's immune system attacks its own tissues.
Yes, and that's where things get really interesting. The researchers found that people with genetic variations in the DNASE1L3 gene are more likely to develop lupus. This suggests that the enzyme might play a role in preventing the immune system from going haywire and attacking healthy tissues.
So how did the researchers go about uncovering all of this? What kind of studies did they do to figure out how DNase 1L3 is involved in both plasma DNA and lupus?
They actually used a three-pronged approach: a human study lab experiments, and then a really clever experiment with mice and gene therapy.
Okay, let's break those down one by one. What did they do with the human study?
In the human study, they compared people with variations in the DNASE1L3 gene to healthy individuals. And what they found was pretty striking. Those with the faulty gene had a completely different plasma DNA profile.
Different in what way? Were the fragments bigger, smaller?
Both, actually. They saw both longer and shorter fragments in the blood of those with the DNASE1L3 variations. Specifically, there were more fragments longer than 250 base pairs and fewer of the typical 166 base pair fragments that are usually quite common in plasma DNA. They even came up with something called a fragmentation index to measure these differences and found it was significantly lower in people with the gene variations.
Interesting. So it seems like DNase1L3 really does play a crucial role in determining the size of those DNA fragments floating around in our blood.
And that's not all. It goes beyond just the size of the fragments. They also found that people with faulty DNase1L3 were missing specific DNA sequences at the ends of their fragments.
What do you mean by specific sequences? Can you elaborate on that a little?
Sure, so imagine you have a long strand of DNA, and it gets chopped up into smaller pieces. Each of those pieces will have a specific sequence of DNA letters at its ends. These sequences are called end motifs. What the researchers discovered was that people with DNASE1L3 variations were missing specific end motifs, particularly those starting with two cytosines, or CC for short.
So DNase1L3 seems to have a preference for cutting DNA near these CC sequences, and when the enzyme is faulty, those cuts aren't happening correctly, resulting in these missing motifs.
Precisely. And it turns out these CC motifs are actually quite common in plasma DNA. The top 6 missing motifs, all starting with CC, normally make up about 10% of all the end motifs found in plasma DNA. This really highlights how important DNASE1L3 is in shaping these fragments.
That's fascinating. So by looking at the size and sequence patterns of these fragments, they could tell who had the faulty enzyme?
Exactly. It's almost like DNASE1L3 leaves its own unique fingerprint on the DNA. And when it's not working correctly, that fingerprint changes.
Okay, so we've established that there's a link between DNase1L3 and the way DNA is fragmented in our blood. But the researchers didn't stop there, right? They wanted to figure out the exact mechanism of how this enzyme works.
You got it. They moved on to some really cool lab experiments to directly observe how DNASE1L3 chops up DNA. They wanted to see exactly what it does to those DNA strands.
All right, let's dive into those lab experiments. What did they do exactly?
They took DNA from a human cell line, but not just any DNA. They used two types: DNA that was still wrapped around proteins inside the cell's nucleus, just like it exists naturally, and then naked DNA, which is pure DNA without any proteins attached.
Interesting. Why did they use two different types of DNA?
They wanted to see if DNASE1L3 could cut DNA in both forms, or if it had a preference for one over the other. And they also wanted to compare it to another enzyme, DNASE1L1, to see if there were any differences in how they chopped up DNA.
Okay, I'm getting a sense that this is where we start to see the unique fingerprint of DNase1L3 emerge.
You're absolutely right. Let's talk about what they found with DNASE1-3 first. When they added DNASE1-3 to the DNA, it was like a molecular chopping frenzy. It cut the median DNA length from almost 2,000 base pairs down to just 153. What's remarkable is that this 153 base pair size is right in the sweet spot for typical plasma DNA fragments. It really suggests that DNASE1-3 is a key player in shaping the fragments we find in our blood.
So it's not just breaking down DNA, it's doing it in a way that creates those specific sized fragments we see circulating.
Right, and to really nail down DNASE1-3's unique fingerprint, they looked at those end motifs, again, the specific DNA sequences at the cut sites. They mapped out the frequency rankings of all 256 possible end motifs and compared them between DNASE1-3 and DNASE1. The results were completely different. DNASE1-3 showed a strong preference for cutting near those CC sequences, whether the DNA was naked or bound to proteins.
So regardless of what the DNA was packaged with, DNASE1L3 had its signature. What about the other enzyme DNASE1? Did it have any preferences?
Interestingly, DNASE1 could only cut the naked DNA, not the DNA that was still bundled up with proteins. And its cutting preferences were totally different. It mainly created motifs starting with a T, not the CC that DNASE1L3 loved.
Wow, that's a stark difference. It really highlights how specialized these enzymes are. Each one has its own specific job and way of working.
Exactly. This specificity is crucial because it ensures that DNA is broken down in a controlled and organized manner. And any disruptions to this process, like variations in the DNASE1L3 gene, can have significant consequences.
Which brings us back to the connection with lupus. Since we're seeing that DNASE1L3 seems essential for proper DNA breakdown, what happens when it isn't working correctly in people with lupus? Could this be contributing to the disease?
That's a great question, and it's one that researchers are actively trying to understand. Lupus is a complex disease, but we know that one of the key features is that the body's immune system starts attacking its own DNA.
So in a healthy person, DNASE1L3 is doing its job, chopping up DNA and preventing it from triggering the immune system.
Precisely. It's like a cleanup crew, making sure that all the DNA debris is properly processed and removed before it can cause any trouble.
But in people with faulty DNASE1L3, this cleanup process isn't as efficient, leading to a buildup of DNA that could potentially trigger those autoimmune reactions in lupus?
That's the current thinking. The abnormal plasma DNA profiles they saw in people with the DNASE1L3 variations suggest that the enzyme isn't clearing away DNA, as effectively potentially leaving the immune system exposed to more of its own DNA.
Okay, I'm starting to see the link here.
Yeah.
But the researchers didn't just observe these differences in humans and in the lab. They took things a step further with a really intriguing experiment using mice and gene therapy.
Yes. They wanted to see if they could actually fix the abnormal plasma DNA patterns they saw in mice lacking DNASE1L3. And to do this, they used a really clever technique.
Can you walk us through that experiment? I'm curious to hear how they approached this.
They took mice that were specifically bred to lack the DNASE1L3 gene, which meant their plasma DNA was all out of whack, just like in the humans with the gene variations. Then they injected these mice with a virus carrying a healthy DNASE1L3 gene.
Wait, a virus? Isn't that dangerous?
Not in this case. The virus they used was specially engineered to act as a delivery vehicle, like a microscopic taxi, carrying the healthy gene directly into the mice. And they targeted the virus to specifically deliver the gene to the liver, using a promoter that ensured DNase3 production, mainly in liver cells.
That's pretty amazing. So they basically gave those mice a working copy of the gene they were missing.
Exactly. And the incredible thing is it worked. The mice that received the DNASE1L3 gene therapy started showing a more normal plasma DNA profile. Their fragmentation index doubled compared to the control mice. And in 2 out of 3 mice, they saw the return of that 166 base pair peak that's characteristic of healthy plasma DNA.
So they were able to partially restore the normal DNA fragmentation patterns just by introducing a functional copy of the gene.
That's incredible. What happened to those CC and motifs? Did they reappear?
Yes. They started seeing those missing CC motifs pop back up in the treated mice. Their plasma DNA fingerprints started looking much more like the wild-type mice, the ones that naturally had a working DNASE1L3.
That's really remarkable. It seems like this gene therapy approach has a lot of potential for treating diseases related to faulty DNA breakdown.
It definitely opens up some exciting possibilities for future therapies. But there was another really interesting finding from this experiment that points to a bigger mystery surrounding DNASE1L3.
Oh, I'm intrigued. What was that?
Remember how we talked about DNASE1L3 being produced by immune cells? Well, in this experiment, they delivered the gene to the liver, not immune cells. and yet they still saw improvements in the plasmid DNA profile.
That's interesting. So even though the enzyme was being produced in a different location, it was still able to find its way to the DNA in the blood and do its job. It seems so. And that's led researchers to believe that DNA's ESE1L3 might be working outside of cells too. It's possible that it's being released from cells, maybe packaged in tiny vesicles, and then traveling through the blood to reach its target DNA.
Wow, that's a whole new layer of complexity. So instead of just sitting inside immune cells, it's going on a little journey through the bloodstream to chop up DNA wherever it finds it.
That's what the evidence suggests. And it raises some really fascinating questions about how this enzyme is regulated and how it finds its target DNA.
It's like we've uncovered one piece of the puzzle only to find that it opens up a whole new set of questions. But it's these questions that drive scientific discovery, right?
Absolutely. And this is just the beginning. As researchers delve deeper into the world of plasma DNA and the role of DNASE1L3, I'm sure we'll uncover even more fascinating insights.
I'm ready for those insights. So we've talked about the human study, the lab experiments, and the mouse model, all pointing to the importance of DNASE1L3. But what's the big picture here? Why should we care about this seemingly obscure enzyme?
That's the key question. What makes this research so compelling is that it goes beyond just understanding how DNA is broken down in our blood. It has implications for diagnosing and treating a range of diseases, including lupus.
Okay, let's talk about those implications. How can understanding DNASE1L3 help us diagnose diseases?
Remember those unique plasma DNA profiles we've been talking about?
Meh.
Well, those profiles could potentially serve as a diagnostic tool.
You mean we could look at someone's plasma DNA and tell if they have a problem with DNASE1L3, which could indicate a higher risk of developing lupus or other autoimmune diseases?
Exactly. By analyzing the size, the end motifs, and other characteristics of the DNA fragments, we could potentially identify those with faulty DNASE1L3 activity. It'd be like having a window into the health of their immune system and their risk for certain diseases.
That would be incredible. It could lead to earlier diagnosis and intervention, which is always a good thing. But what about treatment? How could this research translate into new therapies?
Well, the mouse study with the gene therapy is a great example of the potential here. If we can manipulate plasma DNA patterns by delivering a healthy DNASE1L3 gene, imagine the possibilities.
You mean we could potentially use gene therapy to correct those faulty DNA breakdown processes in people with lupus or other autoimmune diseases?
That's definitely a possibility that researchers are exploring. It's still early days, but the results of that mouse study are incredibly promising. And it goes beyond just gene therapy. Understanding the specific mechanisms of DNASE1L3 could lead to the development of new drugs that target this enzyme, either boosting its activity or inhibiting it in cases where it's overactive.
That's amazing. So this research could have a real impact on people's lives. It's not just about understanding a tiny enzyme, it's about unlocking new ways to diagnose and treat diseases.
Exactly. And it all starts with curiosity, with asking questions and delving deeper into the seemingly mundane world of plasma DNA. Who would have thought that these tiny fragments could hold so much potential?
It's a great reminder that there's always more to discover, even in the places we least expect it. But before we get too carried away with future possibilities, let's go back to the article for a moment. Was there anything else that particularly stood out to you?
You know, there was one detail that I found quite intriguing. Remember how they found that DNASE1L3 could cut both naked DNA and DNA bound to proteins?
Yes, and how that was different from DNASE1L, which could only cut the naked DNA.
Right. But what's even more fascinating is that DNASE1L3 showed that same preference for cutting near CC sequences, regardless of whether the DNA was packaged with proteins or not.
So those proteins, the histones, didn't seem to hinder its ability to find those specific sequences.
Exactly, and that's really interesting because it suggests that DNASE1L3 might have a very precise way of recognizing and targeting those CC sites, almost like a key fitting into a lock.
It makes you wonder how it does that right. What's so special about those CC sequences that DNASE1L3 can find them even when the DNA is all bundled up?
That's a great question, and it's one that researchers are actively investigating. If we could understand the precise mechanism of how DNASE1L3 recognizes and cuts at those specific sites, it could open up new possibilities for gene editing and other biotechnological applications.
It's like unlocking a secret code within our DNA.
Precisely. And it all comes back to the importance of basic research, of exploring the fundamental mechanisms of life. You never know where those explorations might lead.
Speaking of explorations, I have another question about that mouse study. They injected the virus carrying the healthy DNASE1L3 gene directly into the bloodstream, right?
Actually, they injected it directly into the liver.
Yeah.
They were specifically targeting liver cells to produce the enzyme.
Oh, that's right. I'm curious, why did they choose the liver? Couldn't they have delivered the gene to other organs or tissues?
They could have, but the liver is a really interesting target for this type of gene therapy. A highly active organ that produces a lot of proteins that are secreted into the bloodstream. So by delivering the gene to the liver, they could essentially turn it into a little factory for producing DNASE1L3.
That's clever. So the liver is churning out the enzyme and it's getting distributed throughout the body through the blood. Makes sense.
Exactly. And this actually supports that idea we talked about earlier, that DNASE1L3 might be working outside of cells traveling through the blood to find and break down DNA.
This is all so fascinating, but I feel like we've only scratched the surface. The article also mentions another enzyme called DFFB, which is involved in plasma DNA fragmentation. What's the role of this other enzyme and how does it compare to DNASE1L3?
That's a great question. DFFB is fascinating because it acts as the first responder to cell death. When a cell decides to self-destruct a process called apoptosis, it activates DFFB to start chopping up its DNA. But unlike DNASE1L3, which can work both inside and outside of cells, DFFB only works inside the cell.
Okay, so while DNASE1L3 is like a traveling cleanup crew, DFFB is more like an in-house demolition team.
That's a great analogy. And DFFB's role is really important because it ensures that DNA is broken down in a very organized way during apoptosis. This helps prevent the DNA from triggering inflammation and unwanted immune responses once it's released into the bloodstream.
So it's like a pre-cleanup, making sure the DNA is packaged and ready to go before it leaves the cell.
Exactly. And then DNASE1L3 comes along later, possibly both inside and outside the cell, to refine those fragments, giving them their final size and sequence pattern.
It's like a tag team operation with each enzyme playing a specific role in this complex breakdown process.
Exactly. It highlights how interconnected and balanced our biological systems are. Disruptions to this balance, like variations in DNASE1L3, can have significant consequences.
And that's why understanding these processes is so crucial, right? It not only sheds light on the fundamental mechanisms of our bodies, but also opens up new avenues for diagnosing and treating diseases.
Absolutely. By understanding the roles of these enzymes and how they contribute to those unique plasma DNA fingerprints, we can potentially develop new diagnostic tools.
We could look for specific markers in someone's plasma DNA to assess their risk for certain diseases. For example, if we see a low fragmentation index or those missing CC motifs, it could be a red flag for potential problems with DNASE1L3, which could then lead to further investigation.
Exactly. And early detection is always key. The earlier we can identify individuals who might be at risk, the earlier we can intervene, potentially preventing the development of serious conditions like lupus.
And it's not just about diagnosis. This research also points towards new treatment possibilities like gene therapy or drugs that target these enzymes directly.
The potential is huge. Imagine a future where we can precisely manipulate the activity of these enzymes, either boosting their function or inhibiting them as needed to restore the balance in these intricate breakdown processes.
It's exciting to think about the possibilities, but it's important to remember that this is still early-stage research. There's a lot more to uncover and understand before these treatments become a reality.
You're absolutely right. But the progress that's been made is incredibly promising. It just shows how powerful basic research can be, how exploring the seemingly mundane details of our biology can lead to groundbreaking discoveries.
This deep dive has really opened my eyes to the amazing potential of plasma DNA. It's not just cellular debris, it's a treasure trove of information that can tell us so much about our health and guide us towards new diagnostic and therapeutic strategies.
It's truly remarkable, and as research in this area continues to advance, I have no doubt that we'll see even more amazing discoveries in the years to come.
Well, I'm certainly looking forward to those discoveries. It's been a pleasure exploring this fascinating topic with you. I always learn so much from our deep dives.
The pleasure is all mine. And to our listener, thank you for joining us on this journey into the world of plasma DNA. We hope you found it as intriguing as we did.
Remember, curiosity is the key to unlocking new knowledge. Keep asking questions, keep exploring, and stay tuned for more deep dives.